Well, this is really a stunning turnout in honor of the man. Very fitting. It's a real pleasure and a great honor to introduce Frank Solomon to you today. I wrote notes, and I hate using notes, but I thought I needed to make sure that I said what I wanted to say, so I'll try to do a mix and match here. You know, it's interesting introducing someone because you have to really start thinking about what you think of them, right? And that can go a number of places. For me, when I started thinking about what I thought of Professor Frank Solomon, there was this kind of bifurcation. It was the parting of the waters, and I saw there were two threads that I wanted to address with you. The first is the professor as a scientist, and the second is the person behind the professorial garb. I'll try to go both places. I have long ago firsthand knowledge of both of those threads. I met Frank, and we just discussed whether he remembered this. I was very impressed he kind of did, but there was absolutely no reason he should have. But for me, there was. I met Frank at a Gordon conference when I was a beginning graduate student that I'd somehow managed to get myself into. It probably had the name Introduction of Macromolecules into Eukaryotic Cells. Long gone. But for me, it was a seminal experience trying to learn molecular biology. And that was where I encountered Frank's work. In a little movie clip in my mind, Frank and I are leaning against a fence in the sultry New Hampshire summer, out there in the country, discussing beta-tubulin or tubulin and why cells die if there's unpolymerized tubulin. For me, with my, at the time, not so great knowledge of molecular biology, it was a profound understanding that cells were somehow counting what proteins were there and whether the right ones were there. And I thought it was just fascinating and extraordinary that there was this phenomenon that Frank was studying and had insight into. But what sticks in my mind equally to the great science was the tone of the conversation. This was a great, albeit young, MIT professor, and I was a graduate student, but we had a conversation that was a conversation between two people. He listened to my naive questions and answered them thoughtfully and respectfully, and there it was, this kind of depth that I understood scientific discourse could take, the collegial seriousness of really good scientific conversation. It made a very deep and positive impression on me at the time. That's never left. Okay, so who then is Frank Solomon, the scientist? I have to do this part. Frank received his BA in history from Harvard and then switched to the study of life, obtaining his PhD in biochemistry from Brandeis. During his postdoc years, he developed his interest in cell biology and then joined the MIT faculty in 1974, where he began or continued his work on tubulin and the connection between tubulin function and the life and death of the cell when the tubulin is not properly sequestered. His work is seminal, bringing a deep understanding to the field of cell biology in its broadest sense, regarding how the cell counts its protein stoichiometries far beyond the specific proteins that he has studied. Beyond his research acumen, there is Professor Solomon, the unparalleled teacher. He's received numerous honors over and over for his teaching prowess. He served parenthetically as the chair of education for the American Society of Cell Biology. But what I want to tell you is that 3 times he has received the School of Science Teaching Prize, given to the top teacher across the School of Science faculty, a faculty the size of Caltech. He is the only faculty member in the history of the School of Science to have received this prize 3 times. It is a true accolade to his connection with students and to his facility in bringing the subject to his classes in ways that really resonates with the recipients. But part of the reason that Professor Solomon's teaching is so fine is because of the other thread, the person, the professor as person. Frank Solomon, the human being who cares and respects the individual. I've seen him as such in many places, in having the pleasure to serve on committees with him, where I've seen his concern about what is right, and his forthright, clear thoughts are understanding and bring a really extraordinarily useful bent to any proceedings. I've seen him guide the graduate students and the postdocs in their ethics courses, to which I contribute, hitting just the right note of friendliness and humor, juxtaposed with a deep seriousness of the topics at hand. Frank is an extraordinary mentor. He is someone who people can connect with at the level of students and beyond. He cares, he has insight, he is not afraid to share his insight, but he's able to do it in a way that really resonates with the people who are lucky enough to receive it. This all, for me, uncovers the best part of Frank, the human looking for a conversation, a connection with another person whose story he wants to hear. He brought it to our first conversation back in the New Hampshire sunshine. Beyond the stunning intelligence, beyond the biting criticism tempered with humor, beyond the research acumen that has honored the MIT biology department, Frank's humanness has infused our department in the most valuable possible way. It is my honor to be Frank Solomon's colleague and to introduce him to you today.
Hi, thank you for coming. Do I have to sit down, or can I move? You'll be sorry. You're the black one. Not legibly. Thank you, Hazel. So I'm very glad to have a chance to talk a little bit about what this job was like for me all these years. I've noticed that I've watched virtually all the videotapes of the people who came before me. I noticed that people spent a long time talking about how they got here, and so I'll do a bit of that, although I'll make an apology, and then I'll talk a little bit about what it was like to be here as a scientist, and how much I enjoyed this job, how great it was for me. I love being in a lab. I love being responsible for a lab. I love mentoring students. And now that I don't do that anymore, I still love teaching and talking with people. So this has been great. I'll try to bring up some other things that happened along the way. Some things that are important to me now, and I'll bring those up in some form. And then I hope you'll bring up things that matter to you all. Okay, so it's a little tricky for me to talk about how I got here, in part because it's not so linear. The history major, by the way, was the kind of hint. It's been, I think, my path is most noteworthy for its extraordinary bumpiness. And it might seem, after you've heard a little bit about it, that there was no single person behind it, but rather a series of personalities. But I've been evaluated clinically, and it's just the one personality. And it was mine, and it still is. So what I'll do is I won't try to stitch it together into some sort of coherent narrative. Instead, I'll tell you some points and leave out the idiosyncratic and highly personal, if not to say embarrassing parts. Okay, so I was born in Pittsburgh, but we moved to Seattle, which was then a very different place than it is now. I grew up there. Neither of my parents nor my older brother graduated from high school, and everybody actually worked real jobs for a living. And so there was the whole notion of anybody having some sort of life of the mind or life in science was not on the table. So the question is, how did this happen, this strange path? So I had one wonderful teacher in high school, Miss Clark. You may have heard of her. All of my friends from high school, all these years later, still use Miss Clark as a reference point and bring her up when somebody says something moderately biting. She changed my idea of what was education and what it was possible to think about. She changed my idea about politics, about personal courage. She was great. So that was one big influence. When I was a sophomore in high school, in biology class, we did a genetics experiment in which I successfully and irreversibly etherized some flies. So I decided, which made me very interested in genetics, of course, because so I went down to the University of Washington and applied for a job. There was no genetics department then. There were some medical geneticists over in the medical school, and there were 2 geneticists who worked in genetics, as you all would recognize it. But there was no department. They were in a building where the botany and the geology departments were, where the air had not been changed in 30 years, as near as I could tell. And those 2 people were Herschel Roman, who is in many ways the father of, or the progenitor of yeast studies in the States, and David Stadler. And they hired me, of course, with my excellent American history background as a dishwasher and media maker, which was not what I thought genetics was when I killed all those flies, but similar, the autoclaving. So so it was great. That was great. Those 2 men were terrific to me. They had small groups, mainly graduate students, all of whom were terrific to me. When I was done making media and washing dishes, they let me do experiments. I discovered a new form of neurospora, which turned out to be a piece of dust. That paper's never been retracted. So. And so I learned a lot of genetics, and it was a small community. They were interested in literature, they were interested in music, they were interested in politics, and they were interested in science, and they taught me all of them, and it was really, it was a great situation for me, and it opened up a whole set of worlds to me that I never knew about, and never would have known about. Among the graduate students was a woman named Beth Jones, Elizabeth Jones, who actually became a postdoc here with Magasanik, and then was a professor at Case Western and Carnegie Mellon, and who trained many students who have come through MIT, and who was my friend till the end of her life. And she got even with me by grabbing the microphone from the chairman, who was going to introduce me at the first yeast cell biology session in which our lab was presenting work, and regaling the meeting with stories about Mai Tai and spaghetti sauce, which somewhat undercut my seriousness. Anyhow, okay. So I went to Harvard for college, and was completely unprepared for the level of work that, that we were supposed to do. After I figured out a way to stay afloat, I started swimming all over the place, and I had far too many interests to get anything serious done. So I was interested in science and history and real-life politics and literature and what we refer to as stuff, the other parts of life, which I would never bring up in such a serious venue. So in all those areas, I had wonderful teachers there. The biologists were 2. Well, I only took 2 biology courses. One of them was taught in part by a guy named E.S. Castle, who came from a famous family, but he himself was only famous after he died for his work. He was a plant physiologist, and he was great to me and took me seriously and taught me a lot. And the other major teacher I had there was Keith Porter, who was an early inventor of the electron microscope, and it was my second and only, and second of 2 biology courses, and he showed pictures. That's all he did. He would come in, he would show a picture, then he would show another picture, he'd talk about it a little bit, and the pictures were great. They were amazing. They implied a whole set of worlds that we didn't understand at all, and actually all there was then were these pictures. So I always remember those pictures. Porter, it was a huge lecture course. Porter had no idea who I was, but years later I gave a talk at a meeting that he was at, and we met, and we became friends. So, but it's true what Hazel said, I majored in history. Actually, I majored in something called intellectual history, which meant that you didn't have to memorize dates, but you got to read lots of books. And I wrote a thesis, an honors thesis. I was awarded a meh, which is cum meh, which is kind of like with honors, but more like meh. So, and I'm told reliably that my thesis is the only thesis that's not on the shelves of the Harvard History Department. I have no idea why. So I left Harvard and history. It was not a good thesis. So then by a series of mistakes and errors and questionable judgments, I ended up in graduate school at Brandeis in the lab of William Jenks, who science had nothing at all to do with the things that interested me in science. He was basically a physical organic chemist of enzymes. He wanted to know where the electron was at all times. And but I wanted to work in his lab because I thought he was terrific. I thought he was rigorous and that he did gorgeous experiments and that he had the ability to analyze both his own data and data that he didn't know anything about with a kind of illumination that I hoped was transmissible. Meh. So, so I did a thesis with him. He was a very austere person. He didn't talk much. He took to rattling coins in his pocket so that people would know he was coming, like the bell on the cat, because people would talk about him in odd, and it embarrassed him, so he would rattle coins whenever he walked down the hall. It was a little weird. It was like Jacob Marley and Christmas. And, but he was, in the end, a wonderful mentor to me. He taught me science, and then we had lots of other things in common. And we stayed very close until his final illness. Time passes. So my wife and I and our 3-year-old son decided to move to Europe and pretty much stop thinking about becoming a scientist for a variety of reasons. So I had a friend from graduate school who got me an interview with a new institute in Basel, and they hired me, and then so we went, which was, I mean, you just can't figure out why you did some things. And so we showed up, and the director of this institute was somebody named Hubert Bloch, and he brought me into his office, and he said that he was glad to have me and that I could do whatever I wanted for as long as I wanted. That's why I went. And so that did 2 things to me. One was it allowed me to revisit those pictures of porters that I'd seen all those years ago in lecture and start thinking about how to attack them. And the other was, it was the beginning of perhaps the deepest friendship I've had as an adult. So I started working on problems of morphology and motility, primarily from a biochemical point of view, since that's what I knew. I was no longer interested in the electron, but I was interested in where the microtubules were and where the actin was and what the shape of the cell was. And I wanted to do it from a biochemist point of view, and I figured I could mop that problem up in a couple of weeks and then move on to intelligence or the immune system or something. That didn't work either. So it was great. It was kind of virgin territory. There wasn't a huge literature to master. Around that time, a biochemist told me that by entering cell biological problems, I was going to waste clean thinking on dirty systems. And a cell biologist told me that I had to free myself from the shackles of rigor in order always a major aspiration. And, But it turned out that by the time I started doing this stuff, there were already a bunch of scientists, geneticists and biochemists with great rigorous training and very, very smart who were interested in the same problems and were already working in this field. And so both of them were wrong. Where else to put clean thoughts but on dirty systems? And where else to have rigor but in the midst of the fog? So it turned out to be a great time to enter the field. So I got to do the work I wanted to do. We were living in Europe. Things were, it was very nice. And I had we had no intention of coming home, but we had a second child in Switzerland, and some medical issues caused the doctors to tell us that we'd probably be better off going back to the States. Not probably. So I started to look for a real, now I had to get a real job in the States. So I came back and started giving seminars on street corners and, you know, subway stations. It didn't work. But among the places I gave a talk was here. And two professors who were here then, Phil Robbins and Boyce Burge, heard the talk and said, why don't you come here for a couple years, get some work done, and then you'll be able to get a better job in the States, because you're not going to get a good job from Europe. And that fit perfectly with the plan, because of the cities that we wanted to be in for our kid. Boston was one of the three, so it was going to work out fine. So we came back for 2 years, and I got a little work done. And then just as I was about to depart for UCSF, Sal Valeria called me into his office and said, would you like to have a job here? I love San Francisco. So I said, yes, I would like to have a job here. We just figured that it would be better for our kid, and better, you know, regardless of anything else, that it was the right choice for our family, and that we'd stay here for 5 or 6 years, and then when I didn't get tenure, I could leave. So we had a lab from 1976 to 2004, and it was all made possible by those two faculty members. It was very lucky, but it was very generous of them. And then the department hiring me also was great. And we had lots of chances. And this turned out to be a great place to do science. Everybody's really smart. Everybody's very serious about their work. And they'll talk to you. And so our science got much better than it would have been. So during those years, my major scientific partner was Margaret Mogendans, who joined the lab in the beginning and was there until the lab ended. And she was and is, because she now works with Tyler, a paragon of science, both by her ability and by her personality and character. She set the tone for the lab. She set the bar for what constituted good work, and she was great. So we had lots of good graduate students and lots of good postdocs during those years, and I really liked working with them. I also liked doing my own experiments. For a while, I was allowed to do my own experiments. Then people started hiding solution bottles from me. Toward the end, I got to the place where I was allowed to be the third and fourth hand on some experiments, and finally, people started scheduling their experiments when they knew I was teaching. So. I think we had a really good time. I liked working in our lab better than any other lab I ever worked in. I really enjoyed it. I also, if I remember correctly, every experiment worked, but that, I'm not sure that's true. When I came here as a faculty member, I knew something about experiments from Jenks, and I knew something about discovery and excitement from just being around science. But I had no idea how to give a talk. I had no idea how to write a complicated paper. I could write kind of simple biochemical papers. There's the electron, it moved, but that was it, right? And a grant, I had no instruction. So I was really would have been in terrible shape had it not been for a bunch of colleagues, especially David Baltimore, David Botstein, and Malcolm Gefter, who rather roughly slapped me into shape by reading my manuscripts and hearing my talks and saying, and similar comments. So I learned a lot from them, and I don't know how I would have picked it up without them. So I'll tell you a little bit about what we did. We set out to study morphology and motility of cells. Where does the information come from that specifies that those manifestations of differentiated cells? How is that information carried to the cytoskeleton, and then how is it carried out? And we took two approaches. Some were molecular, some were phenomenological. They both satisfied something that was at the heart of my curiosity. Sometimes they worked and touched each other and actually complemented one another, and sometimes they just flew off into the ether. So I'll give you maybe one example of each of those, and then I'll tell you something about yeast, and then I'll wrap it up. The phenomenological was basically, in the beginning, an attempt to try to figure out what were the endogenous determinants of differentiated shape inside of a cell. So trying to isolate a cell from many outside influences. That started off with a series of studies of dividing neurons and asking them to differentiate and looking for common patterns of motility and morphology. And it produced interesting phenomena, but actually no mechanism, although not for want of trying. The first molecular stuff we did, when we entered the microtubule field, the big deal was in vitro reconstitution experiments. So you homogenize the cell, you heat it up, you cooled it down, you added calcium. I think that's how Botstein described it, my entire field, I think. Heat it up, cool it down, add calcium. So it was all based on in vitro reconstruction experiments, and what we tried to do was we tried to figure out a way to isolate microtubule structures as they existed in the cell and look at their components that way rather than other ways. And that turned out to work and gave us interesting discoveries that demonstrated that the same tubulin pool in a particular cell had different associated proteins depending upon what structure it was forming, that the assembled and unassembled pool had different structures, and it turned out to have mechanistic implications. The best place where this all came together was when we started looking at a cell type that nobody really was interested in, except, of course, with my enormous instinct for intellectual history, me, which is the chicken erythrocyte. So I know you all know this, but all vertebrate erythrocytes, except for mammalian erythrocytes, except for camels and llamas, have a marginal band of microtubules at their periphery. And what's cool about that is that those cells look quite different than the biconcave discs that you're used to. They look like rugby footballs that have been squished, creating an equator, and the marginal band is at the equator. And the number of microtubules in that marginal band is the same in every cell for that organism, but different for the next organism. So it seemed like the ideal situation to us to study these kinds of things about specification from inside the cell, because here's a cell that develops in suspension. So we set about to study that. We learned a lot, and we wrote a grant that said that if we can figure out what the molecular components are that specify this marginal band, it's going to lead us to insight into the growth cone of neurons. Despite that, we got the money, and it turned out to be true. We found a protein that that is in this position in the marginal band that somehow links up between microtubules and actin. It does the same thing in the growth cone, and it led to a whole bunch of other phenomenological studies. So that was enjoyable for me. So at some point, I started teaching method and logic with David Botstein, who is a geneticist, and whose view of biochemistry is heat them up, cool them down, and I think the third step is add calcium. And so we started talking and decided that we could collaborate on the yeast cytoskeleton. At that point, the beta tubulin gene had been cloned, but the alpha tubulin, what turned out to be 2 genes, had not been cloned in yeast. So with some joint students, we did that and started doing some other things, which led, then we diverged. David went off in one way and we went off another way. And one thing I will tell you is that it turned out that there were formalisms in the assembly of microtubules, starting with the individual polypeptide chains of alpha and beta, that strongly resembled the balance of components paradigm that Nat Sternberg had worked out for phage morphogenesis. So can you imagine what a great feeling this was, this man who killed all those fruit flies all those years ago becoming a real geneticist again and recapitulating this classic work. And it turned out to be very useful in figuring out, as Hazel said, how tubulin poisons and how it dimerizes and how cells take care of proteins that are iffy and that would kill you if you let them. And that led to working out several steps along a new pathway of folding tubulin and heterodimerization. So all those experiments related back to those pictures of Porter's that I saw as a history major. So let me tell you about 2 things more, and then I'll stop. Shortly after I got tenure and didn't leave, Salva Luria decided that he wanted to resurrect a course that he used to teach in the 60s to graduate students. And this was a literature course for first-year graduate students, but not literature like you think. It's like poems and short stories and essays. And he asked me to teach it with him, and we did. We taught it. And so it was a kind of we didn't actually study my bachelor's thesis in the course because, of course, there's no copy of it available, so that made it hard. But we did lots of things in that course. Wonderful things happened in it. The first-year students were glad to have something other than science to do. I don't know how the faculty felt. We could ask them. It was great. And they got to argue with Salva about things that he couldn't trump them with, although he was pretty fierce. My favorite story was the day that Salva decided that we should teach A Room of One's Own, which is Virginia Woolf's essay about what it means to be an artist and a woman. And I said to Salva, this is a wonderful idea, because you and I, being both women and artists, will be able to represent these points of view very well. He said, he called me several names, cowardly, politically correct, and then some other names, and said, of course we can do this. Who else but you and I can do it? So we told the students that we would meet on Tuesday and at the usual time in the usual room, and 15 minutes before, Salva called to say that he wouldn't be able to come back. Okay, so I just want to tell you one more thing, which matters to me, and then we can talk about anything you want. So at some point, I got involved in graduate training here. I was on the graduate committee, and then people started asking me to do jobs, get coffee, things like that. And as I worked my way up through the coffee, and eventually for some years, I was the chair of the graduate committee here. So that set of interactions, plus getting to talk with a bunch of postdocs, I started taking very seriously issues of training and career structure and things like that. And I got a chance to work on this at the American Society for Cell Biology. And so working with them and with a group called the National Bureau of Economic Research, which is a bunch of economists. Up the street, we did, I learned to read economics. I even speak a few phrases like, where's the bathroom? And I can also say a room of one's own in economics. And we did studies, we interviewed people, we wrote papers. I probably gave somewhere between 30 and 50 talks about this stuff all over the country, and I acted like an advocate for change. And it didn't work. I didn't bring about change. So that stopped. I'm bringing it up today because it still matters to me, and because 6 weeks ago or so, a paper appeared in the Proceedings of the National Academy. It's written by Bruce Alberts, Mark Kirshner, Shirley Tillman, and Harold Varmus, all of who are well-known, highly thought-of scientists. And they decided to go low on the title, Rescuing U.S. Biomedical Research from its Systemic Flaws. So it's a paper that's caused considerable comment, especially in a community that I'm still connected with, which is a community of both biologists and economists who still think about this career structure thing. And so I wanted to tell you a little bit about what I've heard from them. So there was no assigned reading for this course, and so this is not a gloss on the paper, but I just want to bring up this issue in this way. So I've heard from lots of people. One of them wrote and said that the paper basically repeats analyses that were done 15 years ago and makes suggestions that were made 15 years ago. And a second person repeated that and then pointed out that they didn't get cited, which is ironic. So, somebody else wrote to me and said that two of these people, Varmus when he was the director of the NIH and Alberts when he was the president of the National Academy, during the time when this original fluff was made in the late 90s and early 2000s, actually didn't support any changes. And in fact, Alberts took the position that we need lots of PhDs and need to keep training them because they'd be good high school teachers. This is a position held by everybody who has never been in high school or never taught high school students. And so it doesn't really hold. There's a way to turn a PhD into teaching high school, but it's not necessarily the obligatory path. For example, restriction enzyme failure. So then a bunch of other people wrote to me and said we shouldn't be washing our dirty laundry in public. That if we say that the profession is in trouble in some way, that the politicians in Washington will get the idea that we can't take care of ourselves. And another issue that people brought up is that it will depress young people, which is why I brought it up today, depress you. And we shouldn't depress young people because they should be happy and go lucky. Because I think people are worried that we're going to push people away from careers in science. And finally, there's a complaint about the paper, and you can read it yourselves, that it's a bit of a scold. That is to say, it says we should do this and bad, bad, but it doesn't really give systemic approaches to how we're going to deal with this. So I just thought I would tell you what I think now. I think it's great that 4 really prominent scientists that everybody thinks highly of have revisited these issues. Late doesn't matter. The fact that they did it, I think, is a big deal. I think that having a frank public discussion makes everybody feel better. If there are issues in our profession then it's not a good idea for people to blame themselves for things that are going wrong, if in fact there are systemic issues, and so those should be aired. I think it's really important to be candid about what's going on. A previous speaker in this series pointed out that the very high age at which people are getting their first R01 or R01 equivalent from NIH. But then the speaker laid that age to MD PhDs as if it were their fault. MD PhDs are responsible for a lot of evil in the world. I mean, I know some, so, but that isn't true. That statement is false. The NIH splits out PhDs from MD PhDs. So it's just not the case. In 2003, a very prominent, well-known scientist wrote an essay that singled out Shirley Tillman and me for misinterpreting the wind. Because, the scientist pointed out, the NIH budget had just doubled, and so there really wasn't going to be any problem. The number of members of FASEB from the year 1920 to 2000 has gone up exponentially. Even during the Depression, there are no points off the line. So I don't know how much pressure the doubling in fact relieved, given the way the doubling was spent. But the prediction is that if that's what we need, we're going to soon be able to take over the Department of Defense with the NIH. There's just not enough money to keep doubling. So I think this is a really crucial issue. I think the field has to face it. I think that leaders have to lead here. One thing that this article does do, it revisits a point made by the National Bureau of Economic Research ASCB study. And that is that the thing that's different about biology from virtually any other field, life sciences research, is that our training force and our workforce are conflated. So it's very difficult to disentangle how we do business from who we train and how many people we train. So there are responses to that issue, but I feel and I think these four people who have now weighed in also feel that unless the field takes steps to disentangle those two strains in our profession, that we're going to continue to be in trouble. Okay, so thank you. At this point, I'll say anything. You don't know what's in this coffee cup, so. Yeah?
So Frank, how did you get from Seattle to Harvard? Where you mentioned Clark, but it wasn't clear.
That teacher. How did I just wanted to get as far away from Seattle as I possibly could. And so, using my map, oh, it's true. It's how I got it. You mean how did I get in? Is what I think that.
Well, we could give up on that.
Yeah,
but
right.
Rather the issue of coming from a family that did not have that kind of a background.
Right.
Were there difficulties in convincing the family for you to do that, or did you have that support even though they didn't have a background? Just what were the dynamics that allowed you to be able to make that transition?
No, I didn't have the support. But I had an airplane ticket. At Harvard, of course, there's no discrimination between the rich and the poor, but the people who are on financial aid come a week early and clean the toilets for the people who are leaving. But otherwise, you can't tell us apart. Well, that's, I'm sure, the mop, I think. Yeah.
Along that same theme, I know those Brandeis chemists that were very rigorous and proud of their work. How did you get from history over their barrier?
So the first person to ask this question was Jenks's administrative assistant, which was in those days called a secretary. And she said, "Why did you take him?" And Jenks said, "I have never met anybody who knew so little." and then she shared that with me. I have no idea. You know, were Jenks still alive, he wouldn't admit to how either.
So Frank, what's your solution to all the issues that you brought up at the very end?
So let me give a too long answer to that. So I think that the road here is that the whole business of depending upon trainees to do all the work ties you to the training force in a way that, and to its size in a way that will never resolve this problem. It's no accident that most trainees are supported not on training grants and not on individual fellowships, but on research grants. There's a clear reason for that. And so one of the things we argued from the beginning was that there should be a classification of scientists who does experiments, who does work. And it would be stably associated with the laboratory. So there are issues of expense, but there are also issues of expense every time you retrain somebody, because students tend to leave in just 3 years or so. So I think that made sense. I'll tell you, one of the issues, it seems to me, is that we're losing people who would actually love to continue to do experiments if it paid a reasonable wage and had certain kind of stability to it. One criticism of that plan was made by Maxine Singer, who said that we were creating what she called a mommy track. That what you would do is you would take all these people who wanted to have children, be they, I think she meant mommy in the non-gender specific way, although I'm not sure how she would do that. But forcing them into, you know, putting them into this other level of job. But the truth is we're losing people. So I'm not sure that creating some professional track in which people actually do experiments and don't run laboratories and therefore are not dependent upon replicating themselves multiple times in a career for the levitation of their work. Yeah.
Isn't that the model in Europe, though? More research scientists in larger institutes?
That job certainly does exist in Europe, and it exists in some places in the States, too. And you don't want to be in Europe? I think at a certain level it works, right? And I think that the profession has to ask itself, are we providing the training for the jobs that people are likely to want to have and likely to be able to get? And I think that's a crucial question to ask, right? And I think, I told you that one of my respondents, one of my friends said, I'm worried that we're going to discourage young people. I am too, but I don't know when would be worse to discourage them.
Frank, do you think this class of research scientists should work within other people's labs, or should they also be independent?
That was the idea. Is that there would still be PIs, for example, you, but that you would have people in your lab, right, who were with you for years and who contained the culture of the lab, transmitted the techniques of the lab, were basically engaged in the lab, but didn't have to write grants, didn't have to train students. Yeah.
Do you think that program should accept less students?
That this program should accept less students. So we never did send out a questionnaire to programs asking them to volunteer who would like to accept fewer students. But the truth is that some programs are much, 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 much better than others. They give better training, they have better students, and who knows which of those came first. But the way that support is allocated now for graduate students, there's none of that kind of quality control. So should this program accept fewer students? I don't think so. Should there be fewer people entering the training paradigm? I think we have to think about that nationwide. Oh no, I've depressed everybody. Should I tell another Salva story that would cheer you up? Yeah.
What advice would you give young people for finding mentors early in a career?
You mean graduate advisors and postdoctoral advisors?
Or beyond that, like just
mentors.
Because mentors are not always just your advisors, right?
You mean, so at any stage of your career?
Yeah.
Isn't there like this trick that speakers could do when they turn it back to the questioner and say, what would you say? And then I think the questioner gives you a look like the one you just gave me. Is that?
I think you do that role well, so I think that.
Say it again?
I think that you fill a mentor role well, so I feel like, explaining, some people don't reach out to find mentors at first, so.
Yeah.
And then.
So here's, this is not well thought through and more experiential than anything. But so when I have visited other departments, I'm appalled at some of the graduate programs. I'm on the visiting committee of a graduate program, and I was just there last month, and I found out that the students have nobody to talk with, because every time they tell it to one faculty member, believe it or not, the faculty members tell it to each other. So there's no, there are all kinds of safeguards here. So I'm not so worried about the students here. Postdocs I worry about here, and junior faculty. I mean, I think that those are big issues. So when we talk to postdocs in formats, in fora that allow this conversation, I tell them to be on the lookout for people who care about their work and will take an interest in them, and to develop a relationship, right? I have this feeling about the postdoc position that I should have been allowed to design it, but did they ask me? No, right? But I think that's the even in a place like this, which cares about its young people, I think the situation that postdocs find themselves in is really problematic, partly because of the job market, for all kinds of reasons. So, how do you find good mentors? I was lucky, so I recommend that you be lucky.
What did students like about the literature course, and how did it meet its end? And if you had the opportunity to devise,
a bunch of women burned us down. It's horrible. Oh my God. Salva running down the street. I'm sorry, go ahead. Yeah.
If you had the opportunity to design something else non-scientific, what would you want to teach students?
Here.
It's not that book.
Oh, so let's see. So what was the first thing you asked me about?
What did students like or dislike about it?
Oh, they loved it. They loved it. They go and read these things that had nothing to do with chymotrypsin. Can you imagine? They form opinions. I should say that Salva and I had a somewhat different attitude. He would go and read all the critical literature about each thing so that he would know, and I would just read, right? And the kids were too busy, right? So they just read, and we'd talk, and we read great things, and they were touched and moved by them, and it was terrific. It met its end because Salva got sick. What would I teach now? I'm tempted to say I would teach my bachelor's thesis, but I can't. It's so self-serving. I actually loved teaching. We didn't teach, right? I loved sitting around talking about poems and short stories and that one essay. If there was something else, it would be something more social and political now. I mean, some of the things that we bring up in the responsible conduct course could make a year, right? So I'd love to talk about those things and bring in people to talk, and I think it would be great. Yeah.
What advice would you give to students that want to become professor and establish their own lab, and what things throughout your career you wish you would have known before you started?
Oh my God. Wish I would have known. I didn't know anything. So I'm not very good at giving generic advice. I'm glad to sit with people and think about their situations, but I'm not so good at. I think there are other people who, and I think actually lots of the talks in this series, if you listen to how they got here, follow those guys, right? The history thing, no, don't do that. I wish that I'd been taught to write. I wish I'd been. My first grant, Salva read it and said, "Oh, the Encyclopedia Britannica. I love reading the Encyclopedia Britannica," and tossed it in the wastebasket. So that hurt a little, but at least I didn't send it out. I didn't know how to write a grant. There were so many things I didn't know how to do that are part of being a professor. It's not just being brilliant, right, like my colleagues are, but they're also good communicators, and they're thoughtful about strategy and things like that. And I think, it's circular, but I think you learn that from great mentors, among other people, right? Really great mentors, scientific mentors. Yeah.
Do you think new faculty members should be given some kind of special training, whether it be writing, public speaking, or management, or anything like that?
So every junior faculty member is different. For example, on a spectrum, there's me, and then there's the rest of. I start off really in the hole. So it would have helped me a lot if somebody had formally taken me in hand. As I said, they didn't, but one person sent me to another, so David Botstein, and Malcolm Gifter all had things to say to me in language that we can't repeat here, but they were very tough and very critical, and it really helped. Right. But I would have rather learned it another way, like, this is a sentence. So that would have been better. The management thing, I don't know what to say about that. There are junior faculty members who come in here who have enormous instinct for how to talk with people, for appreciating that you don't have one mentorship style, you have mentorship styles that's flexible and that fits with the needs and strengths of each individual student. And then there are other people who've been here for 30 years and don't get it. So I don't know who needs help, right? But it's not the easiest profession in which to ask for help, but there's no, to respond directly to your question, there is no institutional mechanism for lots of those needs. So it's ad hoc and one-on-one, I think. And I think it could be better. Yeah.
All of the problems you discussed at the end.
Yeah.
There's lots of discussion about solutions and what's broken. But my question is, what were the origins? How did this all start?
You mean, how do we get in this fix?
Yeah. Who or what?
Who's responsible?
Thank you.
Mainly Alan Grossman, but. And he didn't used to be, but now he's the acting chair of the department, and it's all his fault. It looked like it was going on forever. There was a time when it was easy. Obviously, it was easy to get a job, easy to get a grant, and easy to get some work done and publish. And it looked like it could expand forever, and it can't. For all kinds of reasons, not the least of which Malthus, rabbits, money, the expense of experiments. for some reason or another, even 18 years ago, when the handwriting on the wall was very clear, the field didn't recognize it and make corrections at a time when it could have. And I think there was time to save the situation then. I think now we're in a deeper crisis. But there was just, that somebody could say, well, but the NIH budget just doubled, so we're okay. It's just no perspective on what's happening. So it's a shame. I can name names, and Alan is not one of them, but thanks.